everybody, welcome back to Biotechnica. Today I have come up with yet another topic from unit number 5, developmental biology. The one of the important topic that I can say that you can definitely expect in your CSIR NET examination, which is shoot apical meristem. We used to call it SAM. So first we'll be dealing with 8 points. So SAM, what is SAM? And then we'll be talking about what are the layers that are actually present in shoot apical meristem. And the third point we'll be talking about what are the genes that actually controls shoot apical meristem formations. And then we'll be talking about certain genes, very specifically we'll be talking about clavata genes, what are the types. And then we'll be talking about what is the correlation between two important genes. And then we'll be talking about how the mechanism actually is regulated or what are the steps that are involved in it. And finally, we'll be talking about the working cycle, which is really important. And finally, we'll be just concluding it by having a point to remember. So first, let's talk about what is a meristem. We already know what is a meristem. Meristems is something which we can say is undifferentiated cells, or we can say it is the cell which rapidly divides. And there are very specifically two types of meristems which we know, which is root apical meristem, which we call it RAM, and the second is shoot apical meristem, which we call it SAM. So today we'll be discussing about shoot apical meristems. So where is this shoot apical meristem is present? This shoot apical meristem is present at the tip of the stem, we can say. So at the tip you can find this one. So let's do that. Suppose if I have to draw a structure, suppose let's consider this is the stem, and at the tip you'll be having this shoot apical meristem which is formed. But how this shoot apical meristem is formed, we are going to talk about it. The next one, what is the purpose of this shoot apical meristem? It is the site of embryogenesis in angiosperms. We know in flowering plants, this shoot apical meristem, or we can say shoot, is the one which is going to be converted into a flower. So a flower is going to be a modified shoot, we can say. So from this flower, we know a reproduction or fertilization will take place, and finally we will get an embryo. So I can say indirectly a shoot apical meristem is the site where embryo is actually formed. Yes. And apart from that, it is the primordia of leaves. Yes. So it can also give rise to leaves, sepals, petals, stamens and ovaries. That is where we will be talking about ABC modeling, where even this shoot apical meristem will develop into a floral organs, we can say, which is involved, like we can say floral organ identity genes, which are actually involved over there. Next, we'll be talking about what are the cell groups that are actually present over there. So let's talk about that one also. So what are the 4 distinct cell groups? So there are 4 different cell groups which are actually present in the shoot apical meristem. So let's talk about that also. Suppose if I'm talking this part, so this area is going to have lot of stem cells. So the shoot apical meristem has, the one is a stem cell. The second is a organizing center. Beneath this, you will find some other cells which are actually present. These cells are called as organizing centers. Yes. Apart from that, you will see a founder cell for organ initiation. So if you want this shoot apical meristem to develop into an organ, you need to have some founder cell. Those founder cells are actually present in this part. So these are the green ones that I'm drawing are going to be the founder cell, or we can say this is organ primordia cells. This is organ primordia cells. So the remaining cells are going to be the cells which are actually present in this area, which are called as the daughter cells of the stem cells. So we have talked about what are the cell groups which are actually present in the shoot apical meristem. So one is the stem cell, the second is the organizing cell which is present beneath, and there are a lot of daughter cells of the stem cells, and there are a lot of founder cells. Yes. Now, if we have to talk about the layers of SAM, what are the layers it actually makes in case of a shoot apical meristem? So let's understand that also. The second point is going to be what are the cell layers in shoot apical meristem. So let's draw all the layers. The outermost layer, the innermost 2 layers are actually present in the shoot tip. So this outermost layer is called as L1, and this second layer is L2, and this third layer is going to be L3. So you can check over the points which is here. L1 is going to be the outer layer, L2 is going to be the second layer. L3 is the third layer. L2 and L3 are internally present. Yes, they are present inside. And every layer is going to have their own stem cells. I told you shoot apical meristem has stem cells which usually keeps dividing and they do not do any function. So they are undifferentiated cells. So each of the layer has its own stem cells, and all these three layers leads to the formation of a stem or many organs. Even floral organs we have already seen. Okay, let's understand what are the genes involved in it, and we will understand what is this outer layer is responsible for. So if we have to talk about the outermost layer is responsible for the formation of the epidermal part. The epidermis will be forming from the L1 layer. The rest two internal layers, L2 and L3, you can see, they are the ones which form tissues that make up a plant body. And even L3 is also responsible for making up the plant body. Yes, so now we have understood what are the layers that are actually present in a shoot apical meristem. So the third point is going to be what are the genes that actually controls the shoot apical meristems. There are three classes of genes that are important for making up the shoot apical meristem. The first one is going to be shoot meristem-less gene, and this gene belongs to Knox family. Yes, it belongs to a Knox family. The second gene is called as Uschel genes. We used to write it as WUS. And the third gene is called as Clavata genes. There are three genes which are actually involved, and let's understand what is the function of every gene that is actually present. So we'll talk about what is the next one. Now we'll be talking about what is the function of the genes. What is the function of STM gene? What is the function of WUS genes? And both the genes are going to play an important role. This STM and WUS gene product, which means the proteins that are produced by these genes, keep the meristem in an undifferentiated state, which means this meristem keeps dividing so that the number of stem cells increases. So if a cell divides, the stem cell number, not the size, the stem cell number increases, and it usually causes the increased SAM formation. The size of the SAM actually increases, or we can say a big SAM. This is very important. So what exactly happened? The role of two genes, which is STM and WUS gene, is to actually make them divide and not to differentiate. So there will be an undifferentiated cells. So the role is to keep them undifferentiated by increasing the number of stem cells by division so that big SAM will be formed there. Okay. What about Clavata genes? Clavata genes is the one which limits the number of undifferentiated meristematic cells, which means this Clavata gene is going to do the opposite action of STM and WUS genes, which means they will decrease the number of undifferentiated cells, which means decrease the undifferentiated cells means they are going to decrease the number of stem cells. So if they decrease the number of stem cells, they are going to form a small SAM, a small shoot apical meristem. So just understand the WUS gene and the STM gene usually make up a large SAM, we can say, whereas this Clavata gene is going to produce a small SAM. This is the normal scenario which usually happens. And since they are going to do a function which is opposite to each other, we can say Clavata gene acts as an antagonist to both the genes, which is STM and WUS. Okay. What about STM going to act alone? Can they do the same function? Yes. Even without the use of or do not depending on the WUS gene, STM can prevent differentiation or maintains the undifferentiated cells or increases the number of stem cells by increasing the big size of the shoot apical meristem. The WUS also does the same thing independent of the STM. But here we'll be talking about what is the role of both of them together and Clavata gene. So just understand STM and WUS is the one which actually will not allow any kind of differentiation. So undifferentiated cells will actually be formed, dividing many cells. So number of stem cell increases. As number of stem cell increases, big SAM is formed. But Clavata does the opposite function, which forms a very small SAM. So to summarize very clearly, we can write a basic things which is STM and WUS together. So STM and VUS are going to maintain maximum number of stem cells. Stem cell number increases. So these stem cells are usually going to be undifferentiated ones. And this increased stem cells, as the number increases, we can say they are going to form a big shoot apical meristem. This is the role. But if we have to talk about clavata genes, CLV genes, this clavata genes decreases the stem cell number. Stem cell number. As the stem cell number is decreased, small SAM is formed. This is the normal functioning of both of these things. The next scenario is, what is going to happen if both of them are actually mutated? Just the opposite will happen. If STM and VUS gene is mutated, small SAM will be formed. If clavata gene is mutated, enlarged SAM will be formed. That is going to be the next point. So let's understand what is going to be the next point. What is going to be the mutant effect of this gene? Let's understand. If the STM gene or the VUS gene is mutated, what exactly can happen? If STM and VUS are actually mutated, Wuschel genes are actually mutated. So as they are mutated, what they can do is, the division cannot be performed as such and undifferentiated cells becomes less. So division is less and undifferentiated cells becomes less. Cell is less, which means the stem cell number is less, correct? Yes, the stem cell number is less. If stem cell number is less, ultimately what will happen? The number of cell is less. So it will form a small SAM or a reduced SAM. A small SAM or a reduced SAM. So here we are saying a mutant STM or VUS causes no SAM or reduced SAM, which is small SAM. But the normal functioning is a big SAM, but here it is mutated, so it is giving a small SAM. Mutation in STM alone result in embryo without a shoot apical meristem. So no SAM will be formed in the embryo. Suppose if we are talking about clavata genes, just the opposite action will take place. In clavata gene, what can happen? Clavata gene, the stem cell, if it is mutated, the stem cell number actually increases. So if the number of the cell is more, then it will become a big SAM, or we can say enlarged SAM is going to form. So to summarize, very simple, we can say a normal condition and a mutant condition. Same is written in the mind map. So the first scenario is if STM and VUS is actually present normally without any mutation, it is going to form a big SAM. This is the first thing. And the second scenario is if clavata gene is in a normal situation, then they usually form a small SAM. But if they are mutated, it's just going to do the opposite action. If mutated STM and VUS or Wuschel gene, it makes a small SAM. And here, if they are mutated, clavata gene is mutated, it forms a big STM, or we can say SAM, big SAM. So this is what we have understood. What is a SAM, a cell layer, and what are the genes that are actually involved here. Now we'll be talking about the rest of the things. Let's go and understand the fourth point. Let's talk about what is clavata genes. This is most important. What is this clavata genes? There are different type of clavata genes that has been observed in Arabidopsis thaliana, very specifically the model organism. In the mutant strain only they just observed. It is seen in Arabidopsis thaliana. What are these clavata genes? So there are 3 genes, and these genes is the one which regulates the size, or we can say the rate of controlling the cell division. There is going to be clavata 1 gene, clavata 2 gene, clavata 3 gene. Clavata 1 gene is going to produce an enzyme, or an encodes for an enzyme which is called as leucine-rich lipids receptor kinases. And it is going to act as a receptor, very important. So clavata 1 gene is going to act as a receptor. What about clavata 2 gene? It is also going to code for an enzyme, but they do not have a kinase domains. They lack kinase domain. This is also going to act as a receptor. So clavata 1 and clavata 2 are going to act as a receptor. What about clavata 3? Clavata 3 is going to act as a molecule, signaling molecule or ligand for both clavata 1 gene and for clavata 2 genes. So let's write this one very clearly. So these two are going to be receptors. And this one is going to be the ligand, a signaling molecule. So now the question for us is, where is this receptors present? We said in a shoot apical meristem, there is layer 1, layer 2 and layer 3. So very specifically in the layer 3, it is actually present. These receptors are actually present. So if we have to talk about this one, these receptors are actually present. This is clavata 1 and this is going to be clavata 2. So clavata 3 will be a signaling molecule. That's what we are talking in case of clavata genes. And we know the function of clavata gene. Now we are going to talk about what are the steps that are involved in this process of formation of SAM, shoot apical meristem. So as stem cell number decreases, so initially what will happen? The stem cell number is going to decrease. As the stem cell number decreases, what will happen is the level of CLV3 or clavata 3 is reduced. So I'll be talking about it. So this, if the stem cell number is less, then if you're talking about this cell, so here what will happen? The gene is actually present. Here we have the Wuschel gene. So if the stem cell number is actually becoming less, it is going to act as a signaling molecule and this will allow the expression of WUS protein. And this WUS protein will be responsible for the production or increase in the number of stem cells. So now the stem cell increased. So initially what we have is the stem cell number is decreased. So when the stem cell number decreases, it's going to signal the DNA or the gene or the WUS gene to express, and this WUS gene is going to express as WUS protein, and this Wuschel protein increases the number of stem cells. Now what's going to happen in the next case? The increase in the number of stem cell is going to promote the transcription of CLV3 genes. So if the stem cell is increased, it will stimulate CLV3 expressions, which means we can say the WUS is actually going to, the Wuschel gene actually helps in the regulation of clavata genes, very specifically. So only when a WUS expression is happening, CLV3 genes will be expressed. So increase in the stem number actually activates or promotes the expression of CLV3 genes. Now CLV3 protein is produced. And now this V3 protein is a signaling molecule. Now it is going to bind to the layer number 3, which has the receptors. And these receptors are called as CLV1 and V2. And this CLV3 is going to bind here. And when they are bound here, what is going to happen? CLV3, a small peptide binds to CLV1 and CLV2 and reduces the expression of WUS gene. If WUS gene expression is reduced, the stem cell number again decreases. So can I now say this CLV3 is actually inhibiting the WUS gene, whereas the WUS gene is positively regulating the CLV3, but CLV3 is negatively regulating the WUS expression. So this is what usually happens in this case. So you can understand when the stem cell number decreases, the WUS expression increases. As the WUS expression increases, this is going to increase the number of stem cells. This increased number of stem cell is going to signal the clavata gene, which is very specifically CLV3 gene, and CLV3 protein is produced. This CLV3 go and bind to CLV1 and V2 present in the layers of the shoot apical meristem, and it is going to decrease the number of stem cells, or it decreases the expression of WUS by decreasing the number of stem cells. So this is the step usually happens in case of shoot apical meristem. Now the question for us is, you might be getting the working cycle, or we can say the same pathway which will be taking in these cases. Let's understand this one. So you can see what's the role of CLV1 and CLV3. This CLV1 and this CLV2 are receptors which is present on the L3 layer. So it is present here. This is CLV1 and this is CLV2. And CLV3 is a protein synthesized and moved towards the L3. So suppose here CLV3 will be produce, which is a signaling molecule. And this signaling molecule comes and binds over this side. But when will the CLV3 will be produced? We already know. When the stem cell, I'm writing stem cell as SE. So this stem cell is going to decrease. That time, VUS expression will increase, and this VUS expression increases the stem cell number. Only when the stem cell number increases, that will stimulate the CLEVATA3 genes to express CLEVATA3 proteins. And this will come and bind to this. As soon as they bound together, here we have VUS gene. This VUS gene, they send a signal, and this VUS expression is going to decrease. Here the VUS expression decreases. So the stem cell decreases again. So now we have understood who is the positive regulator. VUS gene is going to be the positive regulator, whereas the CLEVATA is going to be the negative regulator of VUS gene. So now let's understand what is going to be the working cycle. This is the most important image that you will have in your examinations. So you can see two important things which is actually mentioned here. One is going to be CLV3 and CLV1 and V2. And you have VUS expressions. So we know CLV3 is going to be what? The signaling molecule, or we can say the ligand. And we have the receptor, which is CLV1 and CLV3. So this CLEVATA, as soon as this CLV3 goes and bind to CLV1 and V2, it is going to inhibit the VUS expressions or Wuschel gene expression. But in turn, you can see this Wuschel gene, the arrow mark is put like this, which means Wuschel gene is responsible to positively regulate the CLV3, whereas the CLV3 and CLV1 and V2 negatively regulate the Wuschel genes. So this is what is given. Apart from that, we have to understand an important point, which is the point to remember, which will definitely come in your examination. So let's understand what is this case. Mutant cases. If there are certain cases they will be giving in your examination, what will happen if the CLV1 is mutated, CLV2 is mutated, CLV3 is mutated? So let's understand the first point. Mutant CLV1 will cause big SAM. How it is actually happening? So we know if there is going to be stem cell decreases, then VUS expression or Wuschel expression increases. So this is going to increase the stem cell number. So the stem cell number increases. And if the stem cell number increases, it is going to activate CLV1, or we can say CLV3. And this CLV3 goes and binds to CLV1 and CLV2 receptors. But the question for us is, or the point is given here is, if CLV1 is mutated. All this process are similar, everything is happening perfectly, and CLV3, which is a signaling molecule, is also produced. But it cannot go and bind to the CLV1 receptor. So if they cannot go and bind to this receptor, they cannot inhibit the Wuschel expression. If they cannot inhibit the Wuschel expression, the stem cell number will not be reduced. Stem cell number will not be reduced. If a stem cell number will not be reduced, which means the number is actually increased. So it's going to give you a big SAM. So if a mutant CLV1 is seen, then you will have a big SAM, not a small SAM. Okay. The second point says mutant CLV2. Definitely the same case. If CL1 is mutated, then CLV2 is also going to be mutated. So it is also forming a big SAM. Okay. Let's understand the third point. Mutant WUS will form a small SAM. What does it mean? It is Wuschel gene is actually mutated. So we know the stem cell number is less. So this is going to stimulate the Wuschel expression. But they are talking this Wuschel gene is mutated. If a Wuschel gene is mutated, they cannot increases the stem cell number. So if a stem cell number is not increased, definitely means it is going to form a small SAM. The number is not increased, so you will have only small SAM. The fourth point is mutant CLV1 and mutal Wuschel. So we know now mutal Wuschel is going to give you a small SAM. What will happen if the mutal CLV1 is also present? Definitely CLV1 is the receptor. So we know it's a receptor. So this receptor is also mutated. So what will happen? This itself is going to give you a small SAM only. So a small SAM, or we can say the stem cell number, is decreased. If a stem cell number is not able to increase, they cannot signal CLV3 protein. Whether a CLV3 protein is produced or not, this receptor is mutated. So definitely it's going to be a small SAM. The next one, if you have high CLV3, the fifth point is, if you have a high CLV3, what's going to be the scenario? Let's write it. CLV3 is actually more, which means this has increased in number. So the receptors are actually present, which means the stem cell number has increased, which means it is going to create a big SAM, correct? It's going to give you a big SAM. So as this number has increased, it will go and bind to this CLV1 and CLV2. And as they are bound together, what will happen? They are going to decrease the Wuschel expression. And then the stem cell number decreases. And as the stem cell number decreases, it will form a small SAM, which is a normal function. So this also is showing high CLV. As the number of CLV increases, normal function small SAM is formed. The next one, high CLV3 plus mutant CLV3. It is going to form a big SAM. So we know high CLV is going to give you a small SAM. And mutant CLV1, we already know. Whether you have more number of CLV also, let's take this situation. CLV3 is more. You have many CLV3. But the receptor is actually not active. CLV1 is not active. So what will happen? This is a not active receptor. So this cannot go and bind to this. If they cannot go and bind to this, they cannot decrease the size. So definitely you will have a big SAM. Next, if you have more Wuschel, what will happen? The stem cell number increases. So over-proliferation will be taking place in this scenario. So what we have studied, what will happen in this mutant scenario, and this is the most important part you have to take because this will be asked in our CSIR NET examination. So understand it very clearly. So today we have talked about shoot apical meristem formation. In that we talked about what are the types of apical meristems, shoot apical meristem, and then we talked about what are the layers that are actually involved in it, what are the different type of cells that are actually present, and then we talked about what are the cell layers, layer 1, layer 2 and layer 3, and very specifically we talked about what are the 3 type of genes and what are the functions, and Wuschel and STM genes and clavata genes will act each other against antagonists, we said, and what are the function of all this gene, what will happen if they are in the mutant situation we have seen, and then we have studied about what is the role of clavata gene, and we studied about what are the steps that are involved in it and the working cycle, and finally we have done with the points to remember very specifically the mutant cases. So I hope this video, and I'm very sure that this video is going to be helpful for you. If you really like this video, please like, share and subscribe to our channel, and I'm going to meet you back again in a NET video. Thank you all of you for joining.